Good afternoon, everyone. So welcome to the Biodiversity Network and Leverhulme Center lecture. I'm Dr. Lisa Wedding. I'm an associate professor in geography and PI of the Seascape Ecology Lab. And so I'll be first highlighting the talk coming up next week and then introducing our speaker for this week. So next Friday, we have David Cooper coming, who's the former secretary on the Convention of Biological Diversity, and he'll be reflecting on the biodiversity COPs and talking about what that tells us moving forward. So do join next Friday at 4:15. And now I have the honor of introducing Professor Jo Preston. She's from the Institute of Marine Sciences at the University of Portsmouth. And she's our seminar speaker today. She's an expert in coastal restoration ecology, and her research group focuses on understanding marine biodiversity, ecosystem function, connectivity, and value. So her group applies these scientific insights to inform effective ecological restoration and really focuses on that at the seascape scale. So Professor Preston founded the UK-Ireland Native Oyster Restoration Network and was also one of the founding advisory board members for the European Native Oyster Restoration Alliance. So I first met Jo in 2022, where she co-led the first UK conference on ecological connectivity for temperate seascapes at the ZSL. And so today she'll be talking to us about how understanding connectivity across the seascape can inform nature restoration policy and practice. So please join me in welcoming Jo.
Thank you, Lisa, for the kind introduction, and it's really lovely to be here and to see you all. So yeah, I'm going to go through the sort of the outline of my talk, and in celebration of Lisa Wedding's 5 C's of seascape ecology, I framed it around the 5 C's of my talk. I'm going to start off with giving you a bit of context about where we've got to this science I'm going to present today. And then I'm going to delve into some concepts around seascape restoration and ecological connectivity. And then I'm going to put some meat on the bones and give you some real examples of ecological connectivity across a seascape. Then we're going to look at an example of this using carbon. Everyone's interested in blue carbon. And then I'm going to give some conclusions around what this means for restoring our seascapes. So a bit of context, as Lisa said, the native oyster networks in UK and Ireland were set up in 2017. And as groups, we decided to publish some restoration how-to handbooks. We first of all did the oyster one, the native oyster, and then the government asked us to do the seagrass one, and then it was used similarly to do salt marsh and buds. And the idea of this was to take excellent science and expert opinion and make it democratic to enable communities to start doing restoration for the scale up of our habitats that we've lost. And then we ran the Seascape. These were launched at COP26. And then myself and the ZSL ran the symposium on ecological connectivity that Lisa talked about. Because although I like mollusks, I also like the whole of the coastal habitat. And I really felt that if we were going to excel in our ambitions, we needed to get together and get people working in the separate silos of salt marsh and kelp and seagrass and oysters together to look at how the seascape actually functions. And then in 2022, very end of that, we got funding for the Solent Seascape project, which is the UK's first seascape project from endangered landscapes and seascapes. Now, the power of people and network shouldn't be underestimated. When we first started the oyster networks in 2017, there were 3 oyster projects happening, and they were all in the UK. Roll forward 5-6 years later, there's over 30 restoration projects of the native oyster happening around Europe. And that's due to effective communication of science and practice and knowledge exchange. But we're now halfway through the decade on ecosystem restoration. And all those projects are going up in numbers, we're not going up in scale. And we need to think about why. So this is the depressing doom slide that tells you how much of the habitats we've lost. And I'm sure you probably know most of these. So in the UK, we've lost 90% of our salt marshes, of those remaining, over half are unfavourable. Globally, kelp forest in decline. We've lost most of our kelp in the southeast of England. Seagrass, globally declining, between 44 and 90% lost in the UK. Oysters are in a really bad way. 85% of global oyster reefs and over 95% of European ones have just gone. And now we've just assessed that as collapse using the IUCN red listing ecosystem. Why does this matter? It matters because reversing this loss is essential to achieving these goals. This is an example also of how we need to understand what we've actually lost. So Ostrea edulis, the native oyster, the biogenic reef ecosystems built by this species declined prior to scientific monitoring. Therefore, looking back in the scientific records is no good. We want to find out what it looked like. And so we used historical ecology, going back over 1,600 records over the last 350 years to find out what this habitat actually looked like. And this was led by Ruth Thurston from Exeter and Feline Zerm-Gasson, and I dug in with the biodiversity and some of the historical records. And what you see, these orange dots on the screen show that the average length of oyster reefs were over 100 kilometres, and the areas were often well over 100,000 hectares in scale. We used to have 1.7 million hectares of highly structured oyster reefs spanning for up to 200 kilometres all across the coast of Europe, and they've gone. So why does this matter? Well, nature is essential for human life, but unfortunately the reverse is not true. And it's very much that the planetary crisis, the triple planetary crisis of climate change, pollution and biodiversity loss that humanity has caused and now faces is really played out at that land-sea interface. And when we lose this habitat, we lose the ecosystem services that it provides on which we depend. The clean air, water, climate regulation. And underpinning all of this is the biodiversity. So ecosystem services are just the direct and indirect contributions that ecosystems provide for human well-being and quality of life. And this is a really nice diagram of the services or benefits from the sea, all the different environmental ecosystem services that we benefit from habitats and marine environment. This is from Nature Scott. And so at that conference at ZSL a few years ago, we got together and started off this big study to find out all the evidence of ecological connectivity across the temperate seascape. And one of the first things we did was let's just have a look about how these ecosystem services are delivered across the different habitats. And this is quite a boring table, but it's also quite exciting. So these are all the different ecosystem services that we define as humans, from biodiversity to ocean regulation to food provisioning. And these are the different habitats across here. And you can see where there's dark blue, there's strong evidence that these ecosystems provide those services. And whereas white, there's no data, or light blue, it's expert opinion inferred. And then we align them to all the sustainable development goals and specific targets. And you can see just by looking, having a healthy mosaic of habitats infers multiple ecosystem services across different ecological niches, increase resilience. But how do these relate to each other? We also have some really key knowledge gaps around cultural services and things like Merle. So we started to dig into trying to define some of these things, and we've defined seascape restoration as simply the restoration of multiple habitats concurrently or sequentially to restore functionality, resilience, and connectivity across a mosaic of habitats in a marine ecosystem. The paper's sort of accepted, and major revisions, I finished them, just submitting them next Friday. So accepted-ish in review. So there's a lot of debate around ecological restoration versus rewilding. There's the principles behind it, and these are some of the definitions. People don't really agree. There's not an agreed definition for rewilding, but I don't think it matters, because what we're doing is looking at the wider concept of ecosystem restoration, this spectrum. And sometimes rewilding will be the perfect solution, and sometimes it'll be ecological restoration. Ideally, you don't need to do any of those. Ideally, you reduce the pressure and nature heals itself. That's what we want. So you need to really think about where your system sits in this spectrum and what level of intervention is needed. And this is just a pretty picture about the Solent Seascape project. I'm not going to go into much detail about this other than to flag that it's here. It's the UK's first seascape restoration project. We're restoring 4 hectares of oysters, 7 hectares of salt marsh, 8 hectares of seagrass, the other way around, bird nesting sites, and more importantly, we're doing a large sediment recovery plan to try and take off the pressures, and the University of Portsmouth, the scientific lead. But it's just to say the monitoring we're doing alongside the theoretical work really plays into each other. And it's recently been endorsed by the UN Decade as an action. So, ecological connectivity. Defined here as the unimpeded movement of species and the flow of natural processes that sustain life on Earth. That's the IUCN via the CMS Convention. Or the movement of geophysical, chemical, and biological materials across a landscape or seascape. We took these IUCN definitions and just applied them, given the expert knowledge at these conferences and workshops and meta review afterwards, to interpret them in terms of the marine environment. And it's often ecological connectivity split into structural connectivity, which describes the permeability of the seascape arising from physical characteristics, such as the proximity of or configuration of habitat patches that confers functional connectivity. So it might be habitat stepping stones that enables movement of organisms through the seascape. Much more tangible in, say, like a forest system. And then functional connectivity describes the responses of organisms to this seascape structure and the movements and exchanges that entail. And this occurs in all sorts of ways. Larval and seed dispersal, migratory movements of individuals, populations or species, the transfer of non-living matter, such as nutrients or carbon and sediment and energy from one location to another. So it's quite a big field, really. There's lots of different terms that use, but we've decided to plump for mechanism is a process that facilitates this ecological connectivity, such as the water flow or the swimming or the active filtration. And so because there was a lot of debate behind that simple slide, we decided to bring this together in a framework to give an overview of ecological connectivity in the marine environment and describing how functional and structural connectivity relates, giving some examples of all those things, the mechanisms that through which they are delivered, and the ecosystem services they support. Now, of course, these are all influenced by climate change and anthropogenic pressures and abiotic factors that influence the community. But that's quite dry, and so we decided to make it into a picture, to make it a little bit more tangible, because there was lots of back and forth about what's structural and functional. And because structural connectivity confers functional connectivity, it can often be used as a proxy or an estimate of functional connectivity. And understand the relationship between the structure and the function, once you know that, then you can probably use more easier metrics like structural measures once you understand how it relates to functional connectivity. And so here we've got sort of distance between different habitats, patch size. You've got the ontogenic movement of animals in the orange, such as using nursery grounds, breeding, foraging prey. You've got the mechanisms of swimming and currents. Say here you've got some oysters. They're functionally connected via benthopelagic coupling through their filtration, which increases water quality, and cycling of nitrogen and carbon and phosphates. And then this has an impact on the primary production of seagrasses due to increased water clarity, enabling greater photosynthetic rate. And so these are examples of how these two all concepts come together. So to summarize, why does ecological connectivity matter? Now, what we really try to do is bounce off landscape ecology and learn from that. Whatever field you're in, learn from other fields. And then we've been trying to take the lead from the terrestrial environment and the landscape ecology, which has sort of been going for a lot longer than the seascape ecology. And it's provided a really good operational framework to consider multi-scale questions and the ecological processes, and it can be applied to things like sustainability science. And understanding connectivity is central to seascape ecology. Obviously, all the work that Simon Pittman's done and Lisa Wedding and a new paper coming out very soon on the 5 Cs of seascape ecology. And we need to remember that coastal ecosystems are mosaics of interconnected patches, despite our tendency and desire to just look at single habitats and single types, the coastal ecosystem mosaic. This connectivity is fundamental across these mosaics is fundamental to maintaining biodiversity and functioning of food webs. We've destroyed, fragmented, and degraded our coastal habitats, significantly reducing this ecological connectivity and therefore our resilience and functionality. And this connectivity underpins the delivery of ecosystem services, which we are entirely dependent on for nature-based solutions to deliver our societal goals in the face of the triple planetary crisis. So we decided to really come up with like a not very natty definition of the temperate seascape to try and embody these findings and this information. And so we describe the temperate seascape as a mosaic of interconnecting habitats occupying the coastal environment across time and space. They're ecologically and physically connected by bodies of water through which living organisms, genetic material, non-living matter, and energy flows. This connectivity operates of many scales, from meters to hundreds of kilometers, and we touch on that in the paper. And importantly, it acts as a really dynamic boundary, and not just ecological, but social, marine, terrestrial, and atmospheric processes interact. That's why it's so degraded, but also gives us a fantastic opportunity to restore and enhance coastal ecosystem integrity and resilience for the benefit of people and planet. So we're just going to dig into some evidence of ecological connectivity to put some meat on those bones. So we looked at over 300 odd publications, and this is an example of structural connectivity, but as you'll be rightly pointing out, conferring functional connectivity. So we know that salt marshes, seagrasses, and intertidal mudflat patches are connected by annual and interannual patterns of sediment movement. And so if you mapped out their structural connectivity, what we know is when we remove those biostabilizing habitats, it has a huge increase in suspended sediment loading, impacting primary production of food webs. And then when we dug into the functional connectivity, we looked at around the 3 broad themes of climate, pollution, and biodiversity to mirror the triple planetary crisis. I will say that our data set is really biased or skewed. So this is a map showing the biogeographic distribution and number of sources of evidence for ecological connectivity. You can see that overwhelmingly the evidence comes from the Northeast Atlantic and Northeast, with some in Australia. And we didn't find, through our structural review, evidence in South America. We don't think for a moment it's not there, but for whatever in the biases in the study didn't bring that up. What we did find is huge amounts of ecological knowledge gaps. So here's some examples of functional connectivity relating to nutrients and eutrophication. In California, there was increased shoot size and leaf growth of zostera meadows adjacent to oyster beds due to uptake of ammonia from the oysters excreting. In Chesapeake Bay, when you had oyster reefs compared to no oyster reefs, denitrification, which is removal of biological available nitrogen into nitrogen gas, which increased up to 275%. And only reduced slightly when it's co-located with salt marsh and seagrasses, probably because they did a lot of the job too. Connectivity has implications for carbon production. Classic example, presence of sea otters in kelp forests and their top-down control of grazing of urchins can increase the carbon production up to 12 to 13 times. As I said before, shellfish reefs filter feeding, shellfish reefs and the filtering they do increases light availability and increases primary production of seagrasses, and the production of their feces increases organic carbon storage. And what is exciting, this happens at sometimes ecosystem scales. So healthy seagrass systems can reduce the partial pressure for carbon dioxide, buffering ocean acidification effects at coastal scales, benefiting shell and skeleton building fauna. and when we look at trophic interactions of biodiversity. In New Zealand, the flow of organic carbon from kelp to fjords and inlets cross-subsidised the reef fish density and biomass by the provision of this basal organic matter. Loss of kelp forests linked to 9% loss of macrobenthic communities, shouldn't say micro. And seascapes of connected seagrass, salt marsh and kelp exhibit higher infaunal diversity, higher fishery biomass and higher trophic complexity. So we started to gather these evidences of connectivity. What does this mean for ecosystem service delivery? So we took a subset of all those studies where there was empirical evidence that connectivity enhanced ecosystem service delivery. And this matrix here shows a comparison of the individual habitats, mud, seagrass, salt marsh, oyster reefs, and kelp, compared to the others, and whether connectivity has evidence in increasing ecosystem service delivery. If there's a dot in there, that means there's some data. If there's a blank square, there's no data. Huge knowledge gaps about the relationship between connectivity and ecosystem service delivery. If it's blue, it's a positive relationship. So it mostly enhances ecosystem service delivery, but that is context dependent. Here, the oyster reefs and biodiversity with the seagrass and a little bit the salt marsh, it all depends on your point of view. So we know that when you have oyster reefs next to seagrasses and salt marshes, there's an increase in crabs that are basically feeding on the epifauna that are living on the oyster reefs. The crabs then go and eat the infaunal bivalves. So the relationship depends on whether you're a crab fisher or a clam fisher, whether it's positive or negative in terms of food production and biodiversity. So it's nuanced and it's often site specific, but lots of things for us to explore and find out. So again, a bit dry. So we decided to use a diagram to bring it to life. And this is our tube map of connectivity, where there's evidence that enhances ecosystem service delivery. So the light blue line, wave attenuation by habitats increases coastal protection and sediment stabilization. And we all think we know this, but actually finding the evidence was quite tricky. The yellow line, movement of organic materials and sediment from between habitats increases nitrogen and carbon storage. Salt marshes near seagrasses sequester more carbon and nutrients relative to salt marshes away from seagrasses. The light pink, the animal movement between habitats enhances biodiversity, nursery function, and fisheries production. The green line, benthic chlorophyll is a food source for oysters. Where benthic chlorophyll levels are higher, oysters have greater biomass and their filtration draws down more carbon. Brown, macroalgae drifting into seagrass meadows increases biodiversity associated with seagrass and in adjacent deep sea areas. Grey, infaunal abundance increases with distance from oyster reefs, while the density of large predatory crustaceans is greatest on mudflats near oyster reefs. Dark blue, denitrification mediated by salt marsh increases water clarity in primary production and carbon sequestration in seagrass. And orange, benthopelagic coupling by mussels and oysters increases the availability of nutrients for seagrass growth, thereby increasing seagrass growth, and filtration of oysters reduces incidence of seagrass wasting disease. So summed up in a nice little pretty tube map. So for the last little while, I'm going to give you a few examples of the work we've done looking at carbon. And really digging into some seascape connectivity and flows of carbon. And this was originally part of the main paper, but the editors made us move it, which is always annoying. And we called it How Does Your Carbon Flow? And it's in prep at the moment. So I'm sure you all know that if you want, blue carbon is all the rage because of the potential for us to mitigate the CO2 emissions and sequester down and store carbon. And if you want to buy some blue carbon credits, you want to do a restoration project and sell some credits, you can only trade in the amount of organic carbon that comes from the seagrasses. That's autochthonous carbon. So there's a carbon by the seagrass, the autochthonous carbon is due to photosynthesis, which ends up in biomass and eventually as seagrass carbon in the sediment. Or there's allochthonous carbon, which means carbon coming in from other habitats, terrestrial salt marsh, benthic macroalgae, that comes and stabilizes in the seagrass due to its presence. So you could say it wouldn't be there necessarily otherwise, but it's not derived from that seagrass itself. And stable isotopes are a really well-established tool for working out where this carbon comes from, using the light and heavy isotope ratios of nitrogen 14-15 and carbon 12-13. So Dr. Emma Ward, who's my postdoc, did this work when she was doing her PhD, and we took carbon cores from seagrass meadows in Langton Harbour and in Ryde, and we wanted to understand where the carbon was coming from. And as well as looking at the isotope ratios, you can also look at the relationship between organic carbon, the ratios between organic carbon and nitrogen. And if the signature of these multi-proxies of both your sediment and the plants and the photosynthetic organisms that you expect to be in your sample are resolved enough, then you can use stable isotope mixed models to go and analyze your mixed sediments to work out where the carbons come from. And what we found, first of all, there was a very high amount of allochthonous carbon, which we sort of assumed might happen. And so we applied these stable isotope mixing models to the carbon fractionation data from the isotope analysis. I say we, Emma did this, and these beautiful diagrams. And we found in Ryde, the open seagrass ones, that seagrass was the least important source of organic carbon. And so when we worked out the relative proportion of the suspended phytoplankton matter and the algae, they were far more higher contributors to the overall organic carbon. In fact, only 84% of the carbon came from outside the seagrass meadows. Now, in the blue carbon codes, you can assume 50% of the carbon is organic. And if you're assuming that of a seagrass meadow in the Solent, you're going to completely ending up with double accounting, or just actually ending up with emissions. When we looked at the Farlington Marshes, which is an estuarine location, again, 88% of the carbon is allochthonous, so it comes from outside the seagrass, but the sources were very different. So the sources were largely from salt marsh and from terrestrial grasses and macroalgae. And so it shows you that even over very small scales, the sources of the carbon is quite site specific. And in terrestrial systems, in intertidal systems in the Northeast Atlantic, very little is from the seagrasses themselves. So what we're doing here is we're really undervaluing this allochthonous carbon. It's still being stored in the seagrasses. So as part of the paper, we decided to dig into what were the main donors and receivers across the seascape. The light blue ones are the ones that we've got included in current blue carbon accounting. Yellow ones aren't. And these ones are being considered for carbon accounting methodologies. And the blue lines are the flows between them. And we describe which ones are the mainly sources, receivers or donors. The take-home message is that carbon is multi-directional. Habitats are often both receivers and donors, and it's context dependent. But if you can only accredit the autochthonous carbon in the Northeast Atlantic, so much of it is allochthonous. So how about you just extend the boundary a bit wider and include multiple habitats? Maybe you include the whole seascape, and then it's all autochthonous. So that's something we're thinking about at the moment. So this sums up our application of this to restoration. Seascape restoration is rooted in the understanding that coastal ecosystems are dynamic and heterogeneous mosaics of habitats interconnected through, by water through which living organisms, non-living matter and energy flow. Therefore, the socio-ecological context of a site, habitat configuration, and connectivity between habitat types will shape the outcomes of marine restoration projects. To restore trophic webs, enhance biodiversity and ecosystem functioning, to deliver ecosystem services requires the existence of a healthy mosaic of coastal habitats maintained by the flows that occur between them. Acknowledging this interconnected nature allows for more effective and holistic management, conservation, and restoration strategies. A seascape approach that enhances connectivity and restores structure-function relationships is crucial for successful seascape ecosystem restoration. So then, well, what does this mean in practicality? We did a suite of 7 recommendations to help deliver seascape restoration of coastal habitats. Because what I would say, it's really hard. You need experts in different habitats. You need multidisciplinary collaborations. We need huge amounts of innovation. We need a change in ambition. So therefore, to do that quickly, we need to use networks to facilitate knowledge exchange. We need to actively develop skills to deliver it. We're not doing that in our curriculums at the moment, and create guidance to deliver these habitats at multiple habitats at scale. We need to integrate the best seascape ecology and an understanding of connectivity into restoration planning. We need to develop restoration and site suitability models. We don't have the data for many of those. It might be the great place, but there's no feasibility of getting there. Half the time you have to restore in the most logistically feasible place rather than the optimal ecological place. So we need to devise restoration suitability models as well as site suitability models that take that social, cultural feasibility, practicalities into consideration as well. We need to develop decision tools to inform the order of seascape restoration that takes account for these causal flows between them. If you've got a really polluted system, there's no point restoring seagrasses first. They're the canary in the mine. You want to improve the water quality first. And if you do that by restoring, say, filtering species, you probably won't have to restore the seagrass at all. It shouldn't be a standalone activity. It needs to be part of our climate smart marine spatial planning. And if we're going to deliver our targets, which we're not on target to do at the moment, we need to mature our nature restoration. And therefore, we need to really work at developing innovation alongside our practice, and do that in parallel with an enabling regulation and licensing environment. At the moment, the licensing environment isn't enabling. It's not bad, it's just not designed for this purpose. So we need to change that. And importantly, we can't wait for the data to justify the restoration. We need to monitor while we're restoring to collect the data that justifies more restoration and find what the quantification of these ecosystem services are, how the environment affects it. So if we can align our monitoring agendas with research agendas and policy goals, that will more rapidly increase confidence in our outcomes and also help support nature positive financial markets by quantifying things like denitrification. This is a terrible slide, but I put it in anyway. These are the global and some example of regional policies that are underpinned through seascape connectivity. I'm not going to read them out. There's just a lot of them. The ones in grey are impacted by connectivity, but do not explicitly use the term, all the others do. And so, a couple of my favourite quotes here by Rachel Carson, my hero. In nature, nothing exists alone. And Nelson Mandela, it always seems impossible until it's done. Anthropogenic stresses have diminished nearly all coastal marine habitats in the temperate environment. And globally, almost all marine biomes are impacted by overfishing, pollution, and climate change. To reverse the global trend of biodiversity loss, we need urgent, bold, and effective restoration action. Hopefully, the evidence presented here is striking and clear. To achieve a healthy and resilient ocean, where marine ecosystems are understood, protected, restored, and managed, and to deliver on climate mitigation and sustainability goals, we need to restore at seascape scales, reconnecting the matrix of coastal marine habitats to restore the resilience and functionality of the temperate seascape on which human well-being depends. Depicted in this little pretty picture here. This is a little bit of a plug. We're having another symposium in June called Reconnecting the Seascape. There's some really cheap student prices, and we'd love people to get involved. We'll be running a workshop to try and do something like this, but moving it on. So some of the academics might be interested in getting involved in that. Thank you very much.
Thank you very much. And we'll open up to questions from the floor. I'll kick off with one on the carbon thing there. One way you talked about it is if you have a wider landscape, a seascape, it all becomes autochthonous. I suppose another way is if that carbon wasn't captured by the seagrass, what happens to it? Does it oxidize away? Is the habitat providing a function of lengthening the lifetime of that carbon in the system?
Yes, so I mean, you can't accredit anything that isn't improved by an action. So if you restored a whole seascape and there was an increased carbon due to multiple habitats being restored, then you can increase the boundary. So it still has to align to your restoration actions that show climate mitigation. So to answer your question, yes, because it's stored in the anoxic sediments, it's stored stably for a long time. Sometimes, I mean, mudflats are great stores of carbon as well, but there's much more flow between different habitats and they can be remineralized. Some carbon won't be, no matter what, it's really stable. Doesn't matter where in the seascape it is, it will just stay, not remineralized. So it's really complex. Rachel Dunk, who works at Manchester Met Uni, is really digging into this, and she's we split the paper, and she's leading the how does your carbon flow. But yeah, I think really understanding which parts are stored, sequestered and stored due to the presence of multiple habitats, and what would be there anyway, is a really important question. So we don't overbill the blue carbon. Yeah.
Okay, great. Okay, should we open up to questions, please?
Thanks, Joe. So you're leading the monitoring for the UK's first seascape restoration project. So I was curious, I know that's ongoing, but curious to hear what some of the lessons learned there are so far, or any surprising findings that you've had.
Yes, so we're looking at biodiversity, carbon stocks, fisheries, biodiversity through eDNA, and then looking at actual fish biomass through nets and various other things. We've done a lot of baseline surveys. The two big surprises, I'd say, is we're not expecting very rapid changes in the seagrass or salt marsh. It's going to take 10, 15, 20 years for them to grow and stabilize sediment. What I really notice is how quickly biodiversity responds to the input of structured habitat. So some other work we've done is when you put oysters back into the coastal environment, the biodiversity is exceptionally high and it's completely distinct and it's biogenetically driven. So that happens within around 12 weeks. You get this really complex community with lots of different trophic connections. And we're already seeing a much higher number of big mobile crustaceans of fish on restored oyster reefs than we are on other places. Before we restored the oyster reefs, Crepigilla faunacata, the American slipper limpet, dominated the system and was stopping the recovery of the native oyster because it's been outcompeted by the larvae. We've been putting these cupels out, which are calcium carbonate covered football hats, little things put in the seabed. And what we found that at the restoration site, we've reversed that. We've had no Crepigilla land. It's mostly Ostroedulus larvae. So we're starting to understand where the numbers game is, where the tipping points are. And scientifically, so that's really exciting. So we wrote a paper in 2019 saying we're all doomed. They're never going to come back on their own without active intervention. And to be honest, I wasn't really sure if the active intervention would work by putting oyster reefs in, but obviously it has created a tipping point. So that's really exciting. This is a long answer. I'll make it shorter, slightly longer. eDNA. So we're using eDNA to assess the biodiversity at lots of different scales. So we're doing sort of control, restoration sites, reference sites, lots of reference sites in case the restoration doesn't work. So you get some really good science out of it anyway, but it is working. And I was slightly worried I was going to spend all this money, lots and lots of time, brilliant postdoc was running that. And all the eDNA would just show the same community because it was so mixed. We're using several markers, but actually we're seeing really high resolution. So distinct communities at really quite small spatial scales from the eDNA samples in both water and sediment. So that's one, a huge relief, and two, really interesting. So sometimes you get very distinct communities over like 100 meters or less.
Great, thank you.
Just building on that, I'm wondering what you can say about the temporal resolution of the eDNA data, because how do you know that a fish hadn't just been passing through your restoration site and actually isn't staying there and contributing to the ecosystem functioning?
Yeah. Short answer is we don't. We don't know from the eDNA itself. We just know that that organism was in that water when we collected it or somewhere nearby and a bit of its flesh wandered through. So we don't know that for sure. We are going to do some sort of ground truthing studies, like doing sort of eDNA samples simultaneously at distance to try and answer some of those, one of my PhD students. But what we are doing to get around that is when we, as well as the eDNA, we're doing loads and loads of small fish surveys. So big seine netting to look at all the juvenile fish. And so we've got a list of the species that we know are there because we've caught them and counted them and measured them and biomass, and then we've got our eDNA samples. And this is also a surprise, although you can't infer abundance, you can't get abundance from eDNA, the relative number of sequences reflects the relative abundance in the actual real fish data. And they're pretty much the same. What the eDNA does, it picks up all the cryptic species. So they're like, we caught these gobies, we thought they were all the same species. The eDNA, there were 7 different species. So it sort of picks up the harder to track things. So it's indirect, but that's how we're sort of trying to sort of ground truth it.
Thank you.
It's for the online audience.
Yeah, thank you very much for that very interesting presentation. So I have actually 2 questions. Let's start with the first one. At the beginning, you showed a map, a world map of the evidence of ecological connectivity, right? Was that just based on temperate regions, or was that okay?
Yeah.
Yeah.
So it's just obviously there's like the knowledge in tropical system is far better
Yeah.
than temperate systems because you can see what you're doing. I mean, like, and also if you're a marine biologist, where would you rather work?
Yeah.
But yeah, no, and basically took inspiration from the sort of the tropical literature. It's like there's all this evidence between, you know, corals, mangroves, seagrasses, and we have got a database with some of the tropical stuff left in it, but for this analysis, we just looked at all the temperate systems. So that's why there's no evidence in the tropics, because it's just the temperate data.
Okay. Thank you. Yeah, I work in East Africa a lot, and I was like, oh, there's something missing. But
Yeah,
I think I missed the temperate bit.
Yeah.
The other question is, majority of the habitats are degraded mainly because of land pollution, right? So some of them, seagrass habitats, for example. I did my PhD in Lindisfarne, working on seagrass and macroalgae cover. Do you think it would be interesting or relevant to actually include some land-based components in the whole system to actually reduce those pressures before you actually start doing any restorations?
Yes, and I think I didn't touch on this because on the thing, but like everything ends up in the sea. So if you're not looking at the pathways to it, then it's a really important part of it. And I think everyone's sort of moving to wholescape ecology or wholescape restoration to really looking at those sort of land-sea interactions. I think that's a really important part of managing your restoration projects. But I think as long as you're aware of that, you can put that into your planning. We sort of didn't want to run before we could walk. We thought we'd just start with the connectivity across the seascape, and then yeah. And it's, you know, it's so complex, the water quality, it comes from so many years, and it's wastewater treatment plants aren't up to scratch, and the increased precipitation in temperate systems means that they can't cope. There's massive runoff, and so it's a real problem. So in the Solent, a big part of it is working with all the stakeholders to see if we can do a Solent recovery plan about reducing those pressures that come from the land to the sea. Yeah.
Thanks. Thanks very much, Jo. Maybe more of a specific question, but you mentioned this connectivity decreasing seagrass wasting disease in a specific case. And I was curious about how that mechanism works and if there have been other cases of pathogen decrease because of ecosystem
Yeah.
connectivity?
They don't actually know for sure the mechanism of it. So it's theory that might just be due to the filtration engine of the oyster reefs, meaning that the seagrasses are less stressed because it's higher water quality, they've got less epiphytes, and a higher water quality environment means less pathogens. So I don't think they decided on the actual mechanism, it's just hypothesized. There's also obviously a relationship between biodiversity. Higher biodiversity, you're going to get less pathogenic organisms. So, but I can give you the paper if you want it.
Thanks. You mentioned that when it comes to buying blue carbon credits, there's a focus on this measurement within the seagrass, and you're saying it's really a hyper focus there. That's almost like a snapshot, if I'm understanding it.
Yes. So at the moment you can only accredit. Say you're looking at take your carbon core of the seagrasses. So we've restored the seagrasses. Take a carbon core, and you can only credit for the organic carbon that came from the seagrass. So it's seagrass derived, nothing that's come from outside it. So it's only the seagrass derived organic carbon.
Got it. So all this measurement is kind of focused on a point in time grab that
yes,
incentive is put out there for.
Yeah.
Any ideas, like brainstorm ideas or anything around a way you could maybe measure some sort of or put an incentive around a measure related to stopping some of the pressure, like a preventative method? Because if I'm understanding, you're saying, look, there's this flow of activity that's happening, and you've set a target based on this one isolated element. Any thoughts on?
So I think any restoration activity that means the blue carbon habitat extends as a result of that activity, and you can prove that if you don't do that activity, it doesn't happen, then you can theoretically accredit that.
You so put a target around the activities.
So it could be via, but you have to prove that, I guess, you haven't removed pressure somewhere else. So it has to be, but mostly it's for like planting mangroves or planting seagrass or
got it.
Yeah. So basically saying we've done a restoration activity, we've put the habitat in, as a result of that habitat, more carbon's been stored, and therefore we can sell that carbon as credits.
Much of the barrier to proving upstream activities is probably proving the causal. Is that, okay.
Yeah, and I think that, and I don't really know actually how that's being fed into sort of blue carbon credits and accounting actually, because I mean, you could theoretically say if you stop doing something, and then
I'm just if you could incentivize a different activity that's not just simply the hyper focus of where it is in the system.
You just have to prove that that activity directly led to the increased carbon stocks.
And then you could maybe put a target on the connectivity. It's but I don't know more.
And finding those metrics is quite tricky. Really tricky. That's one thing we might be trying to work out the workshop, what are these metrics of connectivity that show that ecosystem functioning.
Very, very interesting. I like how you tied it to direct incentives of what people look at. Appreciate it.
Just thinking, following up on that carbon one. So for seagrass, it seems like a lot of work for quite a small area, and therefore quite a modest amount of carbon. And so compared to like replanting a forest or whatever, do you find so when you get funding for this, is it because the funders, the companies or whatever, are pulled in by the charismatic offset value of the seagrass and happy to pay the carbon funder as they go? Because actually, if a company was purely doing carbon budgeting, it'd probably say, "Oh, plant a few hectares of forest, do that." So, what is it that pulls them into this project and that restoration?
So I think it would be a really hard sell to get investment to do restoration in the solent based on the carbon sequestration and storage of the seagrasses. There's almost no net gain, and so actually, but salt marshes are really good. So we don't have any funding that's directly linked to blue carbon. I think all our funders are looking at sort of the suite of services, and when people do come and talk to us about we want to invest because of blue carbon, like you have to be very transparent that there's very little blue carbon. What you do get is all the biodiversity. They're brilliant at denitrification, and there's all the other enabling environments. So Yeah, I think the blue carbon is the buzzword, but that's one of the reasons some of the other work that Emma's done about relative contribution of allochthonous and autochthonous of seagrass meadows around the world, and how you can sort of have proxies about how much is that, is to try and make it a bit more transparent, and have better proxies for knowing where you might want to invest. But yeah, I think that in the Mediterranean, the Posidonia beds, you can get carbon up to here, aren't you? And so that's probably where you want to go if you're investing in blue carbon.
Great. Okay. Yeah.
I've worked with seagrass in the tropics, like in Palau, and it was very fickle. Like it wouldn't be where we thought it would be, or it moves year to year. So I was really like surprised to hear you mention carbon like credits associated with seagrass, and my first thought was like that seems like a dodgy investment because the seagrass is kind of fickle. But in temperate environments, is it different? Like, is it more stable? Yeah, just fundamental science.
Yeah. So
question.
yeah, that's one of the big questions to answer around blue carbon. What is the permanence? You know, because they're less likely to burn down than a forest because it's wet, but if it just shifts. And so some of the work we've been doing this in the solar is trying to understand the stability of it. And actually Emma did some work comparing maps from the 1980s to now, and some of the beds are pretty much the same. There's slight shifts around as the channel morphology's changed. But what we also see is there's huge annual variation. So particularly Zostera marina, which is a little bit more subtidal than Nanozostera noltii, it just massively dies back in the winter. So there's huge shifts in biomass, huge shifts in biomass temporarily due to light availability. So I think these are questions we have to answer really honestly, so people go in eyes wide open. And try and say, like, obviously blue carbon's a lever to get people in, but actually you need to understand the reality of the system. But yeah, they can be really stable, and but they also can be ephemeral and pop up and disappear. I mean, the marine environments are highly dynamic, so I think that's something we need to really embrace as well.
Actually, I'll just have one more than just my ignorance of oyster bed restoration. You were saying that initially you had to actively create a habitat structure for that, and then there was a tipping point where at a certain level the reef, the bed can kind of propagate itself and spread.
Yeah.
Happens there.
So we know. So basically, you want to restore the seabed. There used to be highly structured reefs that were highly structured, three-dimensional, for kilometers of scales all around Europe's coastlines, and we've got none of them left, pretty much. Just get the odd oyster now and again. And so what we've done is we've put lots of mulch down, shell and gravel, and then put live oysters on top to try and sort of just very small areas, so trying to kickstart a reef system. The tipping point we've seen is in the larval communities, and we've done a little bit of settlement work, but you can't see anything, so it's quite hard to actually monitor the reefs because it's so turbid. And what we've seen before, the oysters weren't recruiting, they weren't finishing their life cycle because the invasive snippet limper, Cryphidophora carta, was completely outcompeting the larvae and dominated the substrate. And the oyster larvae didn't want to settle on it. There's something about the biochemistry of it. And that paper paved the way for us getting permission to put the reefs down. And what we've seen now, that in the areas where we have the reefs, no Cryphidophora carta settled, and we've got lots of oyster spat settling on these recruitment structures we put down to monitor that, as opposed to in the other areas, we don't see that. So we can show that we're seeing a localized tipping point in terms of enabling it to be self-recruiting.
Right.
And we're also doing general plankton toast to look at the larval abundance in the water overall. So yeah.
Great. Okay, but you would still need to build structure for it together, or does at some levels the structure start to appear?
Well, it's what we're hoping, but it will take years, decades. It will, they're quite slow growing organisms, and in reality it's going to take 10, 15, 20 years you just have to put the reefs in the right place, so we know where the larvae is going to go, and also make sure there's enough substrate that's suitable for them to settle. So we've sort of got two reefs already in place. We're extending them. I'm going to a third reef, so we're trying to get, like, we know how far the larvae go. We've got hydrodynamic models of the solant, so we're trying to sort of do interconnected reefs that also will disperse the larvae to other substrate. But it's a really slow game.
Great. Okay, we'll take one more and we'll wrap up.
Where you were successful in driving investment beside behind like initial oyster projects, that wasn't driven by some sort of credit or something. What was the mechanism that drove successful investment?
So it was philanthropic.
Okay.
And it was the inshore fisheries department came to the university and Blue Marine Foundation and go, we've stopped fishing, but the oysters aren't recovering. We don't know why.
Okay.
And so my job was trying to work out why they're not recovering, and Blue Marine Foundation was trying to get some money to put them in. And so it was just like small projects when we started to see a little bit of success, and then we've got philanthropic funding. And Marks and Spencer's actually funded the first feasibility report, because I think of the huge cultural sort of love of oysters, historically native oyster. So that's unexpected. So
just in general, have you seen much funding come towards projects, or you've seen success through the things where you have credit buybacks or investments or something, or not so much? It's more on these targeted zones.
In the States, in America, because they've been restoring for 50 years now, they've got really good data on the return on investment of how it supports blue crab fishery, nutrient storage and sequestration, how it benefits. They've quantified those, and so that's driving the investment. At the moment, we've quantified almost none of those. So like what my lab and other labs are doing, we're trying to quantify the nutrient assimilation rates, denitrification rates, biodiversity, to try and sort of get those values and the variance of them out there, so we can start to drive some of that. And I think the biggest interest will become through biodiversity and nutrients, because oysters are real powerhouses in terms of nutrients, and also their benefit. They're sort of enabling habitats for vegetated systems, so sort of a proxy blue carbon habitat.
Got it. And you were saying you have to do the data collection at the same time that you're starting that. Okay, I got it.
But like we knew nothing, there's nothing quantified. So we've started to do mesocosm chambers and denitrification values, and we're just starting to get that data out there. So loads of knowledge gaps for people who are interested in that science.
Thank you.
Okay, fantastic. And really inspiring just to see this project in the Solent, and the scale of this connectivity and thinking around along the challenges. There's a long haul, as you say, really fascinating. So thank you very much. We have drinks around the corner if anybody wants to chat informally after this as well. So do come and join us for a drink or a snack. And thank you once again, Jo.